Hello students, my name is Vinay Joseph Silas and I am working as a teaching associate in Department of Agriculture Sciences and Allied Industries at Rama University, Kanpur. So today my topic is the propagation in the horticultural crops. As you know, important topic in aspects of the horticultural crops. So first thing comes in our mind that what is the propagation? Propagation means the multiplication of the plants through sexual and asexual method. In another word, we can say that mother plant giving a birth to a baby plant. This is known as the propagation. Now coming further, the propagation is divided into three parts. First is the sexual reproduction, sexual propagation. Second is the asexual propagation, and the third one is the micropropagation. Now the sexual propagation is mainly the fusion of the male and the female gametes which are present within the ovule of the flowers. Now second one is the asexual propagation. It is done by the vegetative portion of the plants, which can be classified as the following methods. First is the cutting, second one is the layering, third one is the grafting, and the fourth one is the budding. Now coming to the third part, which is the micropropagation. It is done through the tissue culture oblique cell culture techniques, which is large scale in studies. So today we will discuss about sexual and asexual propagation. Now one thing comes in our mind, that why we use asexual propagation rather than to use sexual propagation. The reason is that firstly, through asexual propagation we can get the uniformity, in the flowering and fruiting. Second one is the early bearing, in which we can get the flowers early as well as the fruits. And third one is the increased production. Through asexual propagation we can increase the production of a particular crop. And the fourth one is the disease resistant. Nowadays we can see that the many pests and disease are attacked in the agronomical crops. So just to make the crop resistant, we have to use the asexual propagation method, so that the plants are not damaged by the coming pest or disease. So the first method of asexual propagation is the cutting. As you know from the heading, that the cutting is the cutting portion of the stem, root or leaf of a plant. It means that we cut a portion of a plant, which is the stem, root or leaf, which helps in the propagation, which helps in the multiplication of the new plants. In propagation, in cutting method, when we cut a vegetative part from a plant, it is kept in a favorable condition so that the plants are able to grow. Now the cutting is divided into three parts. First is the stem cutting, second one is the root cutting, and the third one is the leaf cutting. As the heading depicts, that stem cutting means we cut the stem of a plant. The stem is further divided into three parts: the hardwood and the semi-hardwood and the softwood. As the heading depicts, that the hardwood, the branch which we have to take for the cutting, for the hardwood cutting, it should be about 1 year old. And in the semi-hardwood cutting, the branch should be 4 to 9 months old. And in the softwood cutting, the branch should be 2 to 3 months old in the process of cutting. And now about the root cutting. In the root cutting, we take the roots of a plant which is 2 to 3 centimeters thick. In the root cutting, in respect to the temperate fruit, for example apple, peach, pear, plum, if we are practicing the root cutting, the appropriate time for the cutting is the December. For the temperate fruit, it is for the process of cutting, December is the appropriate month for the cutting process. In respect to the subtropical fruits, it is better that we should carry the root cutting in the rainy season. Now the last one is the leaf cutting. In the leaf cutting, as the heading shows, we take the leaf as a vegetative propagated material to produce the another plant, because we take a leaf which is fleshy in nature, because if this is the diagram of a leaf, in the leaf cutting you can see, like in a plant which is known as pathar chata, that a small leaves are grown in the border of the leaves, in the leaf margin, and this portion, if we cut and then plant it to the another field, it can grow as an individual plant, and this process is known as the leaf cutting. Now after cutting, the second method of asexual propagation is the layering. Layering is the development of roots on the stem while it is attached to the mother plant. Now it is divided into 4 parts. First is the tip layering. As I have drawn the diagram here, it shows that the plant is grown, and 1 year old, we select the 1 year old branch and then we dig into the soil, and this portion will, in this portion the roots will germinate. The roots will germinate, and further we can detach this plant, and this plant will be grown as an individual plant. This process is known as the tip layering. Now the second part is the serpentine layering. It is somewhat similar to the tip layering. In the serpentine layering, we use the 2 waves, so that in the one time we can obtain the 2 plants. First, the branch which is under the soil, it will develop the roots, and then we can detach it from the mother plant, and the second branch, if the root has been developed, then we can detach it from this plant. So in this we can obtain the 2 plants from the mother plant. This process is known as the serpentine layering. Now the third is the air layering, which is the most commonly used in guava, kagzi lime and lychee. In air layering process, we take the branch which is 1 year old, and firstly we remove the bark of the stem, which is about 3 to 5 centimeters, and then after removing the bark, we can see a yellowish color structure which is known as the cambium. Okay, then after removing the bark, we will use a moss grass. It is a type of a grass which is helped to retain the moisture. So we will wrap the moss grass around the bark which we have removed, and after that we will tie it from the upper end and the lower end with the help of the polythene sheet, so that the air doesn't reach into the affected cambium, because once the air enters the cambium, the fungus infection will grow. So in order to make the air layering process successful, we will tie it from both the ends. Okay, and then we will wrap the moss grass with the polythene sheet. And one thing more, the polythene sheet should be of 200 to 300 gauge, and after 20 to 25 days you will see a root is developed in the moss grass, and when you will remove the moss grass, you will see that the cambium has developed the root, and when the root is developed, we will detach it from the mother plant and we can grow this part as an individual plant. This process is known as the air layering. Now the fourth one is the mound layering. Mound layering, in the mound layering process, we will head back the plant 15 centimeter above the ground. And then we will head back the plant, and then we will make a heap. We will make a heap of with the help of mud, so that the roots will develop from here. And then if we destroy, we will cut it, and then we can grow it as an individual plants. This process is known as the mound layering. So the third method of asexual propagation is the grafting. Grafting is defined as joining the two parts of a plant to grow as an individual plant. So the two parts is, the first one is the sign and the second one is the rootstock. Now what is sign? Sign is the upper portion of a plant, and the root is the lower portion of the plant, which is have the rooting system. So the first part is the enarching grafting. Now what is enarching grafting? In the enarching grafting, we take the rootstock and the sign. Both in the rootstock and the sign, they are both in the soil, and they are attached with the help of the polythene sheet. And when the sign gets attached with the rootstock, then it is cut from the mother plant, and this process is known as the enarching. Now the second one is the grafting, veneer grafting. In veneer grafting, this is the rootstock, and the rootstock we take should be 8 to 12 months old. It should be 8 to 12 months old. In the veneer grafting, we make a V-like structure in the upper portion of the rootstock, and we take a sign of a particular variety, and in the lower portion we make a V-like structure in the lower portion of the sign, and we will insert. And one thing more, the diameter of the sign and the rootstock should be similar. If the diameter of the rootstock and the sign is dissimilar, the veneer grafting will not take place. The grafting, or we can say that the union of the cambium will not take place. In veneer grafting, it is very much important that one should take in mind that the diameter of the rootstock and the sign should have the same diameter. Now when we cut the sign, the lower portion like this, then we will insert it in the rootstock, and with the help of the polythene sheet we will tie it with the help of the polythene sheet, and after tying it we will cover it with the polythene cap, so that the water does not enter into the affected part, because once the water gets entered in the cambium, the fungus will grow, and which will result in the failure of the grafting. So after the grafting, we will tie this portion with the help of polythene sheet, and we will cover it with the help of polythene cap, and this is the polythene cap. And after 2 to 3 weeks, you will see, when you will remove the polythene sheet, then you will see that the union of the rootstock and the sign has been completed. Now this process is known as the veneer grafting. Now the third part is the cleft grafting. In the cleft grafting, the rootstock is thicker than the sign, and it is done in the top working. First of all, we should understand what is top working. For example, this is a tree, and one of its branches has been diseased, or it is affected with the pest. So we will take that branch, and we will cut that branch from the main tree, and then we will take the sign of the another variety, and then we can do the grafting process here only, and then we can see that and then we will see that in the sign there is a development of buds, and when the buds will arrive, then we can say that the cleft grafting has been done successfully. Now, this process is known as the cleft grafting, and this cleft grafting is carried out in hazelnut and the walnut.